So the title of my talk is, What is that slime? Well, SLIME stands for the Salerno Laboratory of Integrative Microbial Ecology, and that's where I am here at the Potomac Science Center. But seriously, about slime, if you've ever picked up something that was floating in a pond or river, it was probably slimy. That slime is the community of microbes that colonize that surface. Although some microbes are freewheeling and solitary, others will spread across the surfaces and form an interwoven mat or biofilm. Some microbes will do both, and depending on the conditions, some individuals will break apart, float off, and they'll go form new colonies. What that slime is, though, that slippery, slimy feel on a piece of plastic in the river, or maybe the plaque on your teeth, it's from the microbe's extracellular matrix, which is a fancy way of saying the exterior surface of the microbes. It can be slippery from the polysaccharides and proteins that glue the microbes together, but they also shield and protect them from the environment. During the summer of 2019, I was participating in a summer team impact project at the Potomac Science Center. While I was working on identifying the microbial communities in the water at different sites in Cameron Run and Hunting Creek in Alexandria, other students were collecting and analyzing microplastics in the area. We didn't have a good system for analyzing the microbial communities and what we found because we didn't know anything about their provenance. We didn't know where they came from, what they were, how long they'd been in the river, how long or even if they were on land first, or anything else. Even if we did know some of that information, we wouldn't be able to easily compare it to the other samples. What we really needed was a way to control for these different variables. After thinking about it for a while, this is what I came up with. What if I were to take some common types of consumer plastic, make them microplastics, and then find a way to put them in the river, but keep them from actually getting loose and becoming pollution? And then I wondered if there'd be any difference in what grew on and colonized the different types, and would that change over time? So here's what I did. I collected 8 different kinds of plastics from my own recycling bin. A grocery bag, a milk jug, styrofoam packaging, plastic foam packaging, a water bottle, plastic film from those little airbag packing things, and some clamshell packaging. That was from the heavy-duty shears that I bought to cut it all up. I drilled some holes in a piece of PVC pipe to make little curls, and another common microplastic material is polypropylene, but I didn't have any. So I went and bought 1 foot of polypropylene rope at Lowe's and I cut it up. After cutting it all up, I got 20 mesh bags, filled them with my 10 different sample types, making 2 sets. 10 of them I zip-tied to a tow chain so they'd stay on the river bottom. And the other set I zip-tied to some buoys and some polypropylene rope, which also floats. These 2 replicates were then stretched between 2 different anchors, which kept them in place in the Occoquan River, just off the dock of the Potomac Science Center. We deployed this double-stranded experiment perpendicular to the shore, to let each of the samples be exposed to the exact same conditions of the river, the flow up and down with the tides. We took samples of the water, the mud at the bottom, and also the vegetation on the day that we deployed this. And we did the same thing every time we took samples, which was after 1 week, 3 weeks, 5 weeks, and then 8 weeks. In a related use of these samples, we shared what we collected with Dr. Foster, who I'd call an environmental chemist. He has his lab here at the Potomac Science Center, and another student is looking at what organic pollutants these microplastics are collecting from the environment. And spoiler alert, I think it was a styrofoam. It seems to be like a DDT sponge. It's collecting concentrations of DDT that are thousands of times higher than in the water surrounding it. After collecting the samples, we put small amounts in these tiny vials that have garnet shards inside. When they're put into what looks like a paint shaker from Home Depot, the garnet and the microplastics get shaken up together and the biofilm gets torn apart. The cells break apart and the DNA comes out into the fluid in the vial. After a series of about 20 different steps in the lab designed to separate it out and collect it, we finally have DNA samples. We cut out a section of the DNA, just a specific region of microbial DNA, and then we sent it off to a lab to be sequenced. Sequencing is the process that reads the DNA snippets and puts the actual code, the sequence of nucleotides, into a file or document that we can then analyze with a computer. Once we had all that information, we could compare all the samples to each other by substrate or plastic type, whether it was floating, submerged, and how long it had been in the river. So, what did we find? Well, the amount of data is daunting. From 105 samples, we had over 2.5 million strands of DNA that were sequenced. These were sorted into just over 50,000 different features, which are groups that are extremely similar and would come from the same or very closely related species of microbes. Here's the distribution, and the median and the mean were both at about 25,000 features per sample. When we match these sequences to a database of known microgenomes, we get a hit, and then we can make an identification. Some can be matched to the family, genus, or even down to the species. This bar plot shows the breakdown of each microbe found in each sample. We'll come back to this in a minute, but this is what I can't wait to see as soon as we get our data back. Let's start by looking at the differences in the environmental samples first. This is a visualization of the difference in the microbial communities between the sediment on the left, the vegetation in the middle, and the water on the right. This is scaled by the Shannon Diversity Index, which is a scale that combines not just the number of different species, but also what the relative populations are of each of those species. So higher is more diverse, and we can see the range across all the sample times over 2 months combined together. This isn't measuring what they have in common, but how many and how many different kinds of members are in each community. When we combine all the samples together by type of plastic, we see they all fall within the water on the far left and sediment, the one that's the highest over here. Even on this scale, we can see that there's variation across the different substrates or different types of plastic. Let's look at the individual samples and see if we can identify some of these differences in the community makeup. While we're looking at the phylum level, we see that the most abundant across all the samples are bacteroides, and which are these across the top. This indicates the presence of fecal material in the water, which is what we find. Some studies have used the individual species of bacteroides to determine what animals the feces came from. The spikes here, they're from the water samples. The next most abundant type is cyanobacteria, found most predominantly in the vegetation samples. Among the floating samples, we can see this increase over time, and notably, these are in very low abundance in the submerged samples, which you can see here. One that goes in the opposite direction is this delta proteobacteria, which you can see is abundant in the sediment samples over time. And then initially it appears in low levels of abundance in the submerged and floating samples, but then over time it rises in abundance with the submerged samples having higher levels than the floating samples. Here's the water, sediment, and microplastics, each with their own color. The sediment are in the lower left in dark brown. The water are the group of the blue ones. The floating samples are up here in light blue, and the bottom samples are in light brown. Notice how consistent the sediment samples were over time, and the water samples for this group here. Now, look what happens when I rotate this to see the changes over time. We have the initial environmental samples here, and then the samples that were taken after 1, 3, 5, and 8 weeks. While they all had similar microbial communities after 1 week, here, look how this changes over time. The floating samples continue off here, and the bottom samples also diverge, with one in particular coming very close to matching the community members and relative population of the sediment. We can also see that while they were initially close together, they all separated over time. Each plastic type was not identical in what communities it formed over time. So which one was that came so close to the sediment sample? In this one, I highlighted that one plastic type in red so we can see how it shifts over time. This one was the clamshell packaging, and not only was the submerged one most similar to the sediment by the end, but of all the floating samples, it was also most similar to the sediment by the end. This is definitely something we'll want to look at further, although perhaps we'll be looking at why the others weren't like this one. Another thing we'd like to investigate further is what explains the differences in the same water bottle sample that was treated 2 different ways. One set I cut up into strips, and the other set, after I cut it into strips, I briefly put it in a blender so it would fray and damage the edges. Here's that same sort of plot with just these 2 different treatments. The square is the one that were cut, and the star were the ones that were frayed. Notice that while they were all very close to each other after the first week, the bottom samples diverged more widely than the floating ones. Whatever it was that made them so different at the beginning, though, after 8 weeks they were more similar to each other than the floating ones were. It was Heraclitus that said, "No man can step in the same river twice. It's not the same river, and it's not the same man." Well, we've seen in this river that not all microplastics collect and develop the exact same biofilms, even when we deploy them at the same time. Because the river is constantly changing, we wanted to see what the relative differences were between the different types of potential trash and pollution that we would be seeing over time. Now, not everything goes as you'd expect when you're doing field work. You don't always know what's going to be going on in the world around you either. Our plans for continuing this project have been interrupted. More samples have been collected that have not yet been analyzed, and we're hoping that we'll be able to get back out into the river this summer and collect samples at the 1-year mark. And don't worry, the foam was in a waterproof case.